to read the chapter 16, Chemistry in Everyday Life. By now you have learned the basic principles of chemistry and also realized that it influences every sphere of human life. The principles of chemistry have been used for the benefit of mankind. Think of cleanliness, the materials like soaps, detergents, household bleaches, toothpaste, etc. will come to your mind. Look towards the beautiful clothes, immediately chemicals of synthetic fibers used for making clothes and chemicals giving colors to them will come to your mind. Food materials, again, a number of chemicals about which you have learned in the previous unit will appear in your mind. Of course, sickness, disease remind us of medicines, again, chemicals, explosives, fuels, rocket propellants, buildings, and electronic materials, etc., are all chemicals. Chemistry has influenced our life so much that we do not even realize that we come across chemicals at every moment. That we ourselves are beautiful chemical creations and all our activities are controlled by chemicals. In this unit, we shall learn the application of chemistry in three important and interesting areas, namely medicines, food materials, and cleansing agents. Drugs are chemicals of low molecular masses, approx 100 to 500 atomic unified mass. These interact with macromolecular targets and produce a biological response. When the biological response is therapeutic and useful, these chemicals are called medicines and are used in diagnosis and prevention and treatment of disease. Most of the drugs used as medicines are potential poisons. If taken in doses higher than those recommended, use of chemicals for therapeutic effect is called chemotherapy. Drugs can be classified mainly on criteria outlined as follows. On the basis of pharmacological effect. This classification is based on pharmacological effect of the drugs. It is useful for doctors because it provides them the whole range of drugs available for treatment of a particular type of problem. For example, analgesic have pain killing effect, antiseptics kill or arrest the growth of microorganisms. Second one is on the basis of drug action. It is based on the action of a drug on a particular biochemical process. For example, all antihistamines inhibit the action of compound histamine which causes inflammation in the body. There are various ways in which action of histamines can be blocked. You will learn this in the section 16.3.2. Third one is on the basis of chemical structure. It is based on the chemical structure of the drug. Drugs classified in the way share common structure feature and often have similar pharmacological activity. For example, sulfonamides have common structural feature. Given below, this is the structure of sulfonamide. And the next one on the basis of molecular targets. Drugs usually interact with biomolecules such as carbohydrates, lipids, proteins, and nucleic acids. These are called target molecules or drug targets. Drugs possessing some common structural features may have the same mechanism of action or targets. The classification based on molecular target is the most useful classification for medical chemists. Drug target interaction. Macromolecules of biological origin perform various functions in the body. For example, proteins which perform the role of biological catalyst in the body are enzymes. Those which are crucial to communication system in the body are called receptors. Carrier proteins carry polar molecules across the cell membrane. Nucleic acids have coded genetic information for the cell. Lipids and carbohydrates are structural parts of cell membrane. We shall explain the drug target interaction with examples of enzymes and receptors. Enzymes as drug targets. First one is catalytic action of enzymes. For understanding the interaction between a drug and an enzyme, it is important to know how do enzymes catalyze the reaction. In their catalytic activity, enzymes perform two major functions. First function of an enzyme is to hold the substrate for a chemical reaction. Active sites of enzymes hold the substrate molecule in a suitable position so that it can be attacked by the reagent effectively. Substrates bind to the active sites of the enzyme through a variety of interactions such as ionic bonding, hydrogen bonding, van der Waals force, or dipole-dipole interaction. The diagram given here shows, first one shows enzymes which have an active site, second one is a substrate, and the third one shows how the enzyme hold the substrate. The second function of enzyme is to provide functional groups that will attack the substrate and carry out the chemical reaction. Drug enzyme interaction. Drug inhibit any of the above mentioned activities of enzyme. These can block the binding site of enzyme and prevent the binding of substrate or can inhibit the catalytic activity of enzyme. Such drugs are enzyme inhibitors. Drug inhibit the attachment of substrate on active sites of enzyme in two different ways. First one is drug compete with the natural substrate for their attachment on the active sites of enzyme. Such drugs are called competitive inhibitors. Active site of the enzyme is given. There is a substrate as usual and the drug. The next diagram shows how the drug inhibit the enzyme substrate. Some drugs do not bind to the enzyme active site. These bind to a different site of enzyme which is called allosteric site. These binding of inhibitor at allosteric site changes the shape of active sites in such a way that substrate cannot recognize it. If the bond formed between an enzyme and an inhibitor is a strong covalent bond and cannot be broken easily, then the enzyme is blocked permanently. The body then degrades the enzyme inhibitor complex and synthesizes the new enzyme. Receptor as drug target. Receptors are proteins that are crucial to body's communication process. Majority of these are embedded in cell membranes. Receptor proteins are embedded in cell membrane in such a way that their small part possessing active sites project out of the surface of membrane and opens an outside region of the cell membrane. In the body, message between two neurons and that between neurons to muscles is communicated through certain chemicals. These chemicals, known as chemical messengers, are received at the binding site of receptor proteins. To accommodate a messenger, shape of the receptor site changes. This brings about the transfer of message into the cell. Thus, chemical messengers give message to the cell without entering the cell. There are a large number of different receptors in the body that interact with different chemical messengers. These receptors show selectivity for one chemical messenger over the other because their binding sites have different shape, structure, and amino acid composition. Drugs that bind to the receptor site and inhibit its natural function are called antagonists. These are useful when blocking of message is required. There are other type of drugs that mimic the natural messenger by switching on the receptor. These are called agonists. These are useful when there is lack of natural chemical messenger. Therapeutic action of different classes of drugs. In this section, we shall discuss the therapeutic action of a few important classes of drugs. Antacids. Overproduction of acid in the stomach causes irritation and pain. In severe cases, ulcers are developed in the stomach. Until 1970, only treatment for acidity was administration of antacids such as sodium hydrogen carbonate or a mixture of aluminium and magnesium hydroxide. However, excessive hydrogen carbonate can make the stomach alkaline and trigger the production of even more acid. Metal hydroxides are better alternatives because of being insoluble. These do not increase the pH above neutrality. These treatment control only symptoms, not the cause. Therefore, with these metal salts, the patient cannot be treated easily. In advanced stages, ulcer become life-threatening and its only treatment is removal of affected part of stomach. A major breakthrough in the treatment of hyperacidity came through the discovery according to which a chemical histamine stimulates the secretion of pepsin and hydrochloric acid in stomach. The drug cimetidine, that is tegamet, was designed to prevent the interaction of histamine with receptors present in stomach wall. This resulted in release of lesser amount of acid. The importance of drug was so much that it remained the largest selling drug in the world until another drug, rentidine, was discovered. Antihistamines. Histamine is a potent vasodilator. It has a various function. It contracts the smooth muscles in bronchi and gut and relaxes other muscles, such as those in the walls of fine blood vessels. Histamine is also responsible for nasal congestion associated with common cold and allergic response to pollen. Synthetic drugs brompheniramine, dimetapp, terfenadine, celandine acts as antihistamines. They interfere with the natural action of histamine by competing with histamine for binding sites of receptor where histamine exerts its effect. Now the question that arises is, why do above mentioned antihistamines not affect the secretion of acid in stomach? The reason is that anti-allergic and anti-acid drugs work on different receptors. Neurologically active drugs. Tranquilizers. Tranquilizers and analgesic are neurologically active drugs. These affect the message transfer mechanism from nerve to receptor. Tranquilizers are a class of chemical compound used for the treatment of stress and mild or even severe mental disease. These relieve anxiety, stress, irritability, or excitement by inducing a sense of well-being. They form an essential component of sleeping pills. There are various types of tranquilizers. They function by different mechanisms. For example, noradrenaline is one of the neurotransmitters that plays a role in mood changes. If the level of noradrenaline is low for some reason, then the signal sending activity becomes low and the person suffers from depression. In such situation, antidepressant drugs are required. These drugs inhibit the enzyme which catalyzes the degradation of noradrenaline. If the enzyme is inhibited, this important neurotransmitter is slowly metabolized and can activate its receptor for longer periods of time, thus counteracting the effect of depression. Iproniazid and phenelzine are two such drugs. Some tranquilizers, namely chlordiazepoxide and meprobamate, are relatively mild tranquilizers suitable for relieving tension. Equinil is used in controlling depression and hypertension. Derivatives of barbituric acid with vernol, amytal, nambutal, luminal, and sacchinal constitute an important class of tranquilizers. These derivatives are barbiturates. Barbiturates are hypnotic, that is, sleep producing agents. Some other substances, such as tranquilizers, are Valium and serotonin. Analgesics. Analgesic reduce or abolish pain without causing impairment of consciousness, mental confusion, incoordination, or paralysis, or some other disturbances of nervous system. These are classified as follows. 1. Non-narcotic, non-addictive analgesics. Second one, narcotic drugs. Non-narcotic, non-addictive analgesics. Aspirin and paracetamol belong to the class of non-narcotic analgesics. Aspirin is the most familiar example. Aspirin inhibits the synthesis of chemical known as prostaglandins, which stimulate inflammation in the tissue and cause pain. These drugs are effective in relieving skeletal pain, such as that due to arthritis, and these drugs have many other effects such as reducing fever, antipyretic, and preventing platelet coagulation. Because of its anti-blood clotting action, aspirin finds use in prevention of heart attacks. Narcotic analgesics. Morphine and many of its homologs when administered in medical doses, relieve pain and produce sleep. In poisonous doses, these produce stupor, coma, convulsions, and ultimately death. Morphine narcotics are sometimes referred to as opiates since they are obtained from opium poppy. These analgesics are chiefly used for the relief of postoperative pain, cardiac pain, pains of terminal cancer, and in childbirth. Antimicrobials. Disease in human beings and animals may be used by a variety of microorganisms such as bacteria, virus, fungi, and other pathogens. An antimicrobial tends to destroy, prevent, development, or inhibit the pathogenic actions of microbes such as bacteria, antibacterial drugs, fungi, antifungal agents, virus, antiviral agents, or other parasites, antiparasitic drugs selectively. Antibiotics, antiseptics, and disinfectants are antimicrobial drugs. Antibiotics. Antibiotics are used as drugs to treat infections because of their low toxicity for humans and animals. Initially, antibiotics were classified as chemical substances produced by microorganisms, bacteria, fungi, and molds that inhibit the growth or even destroy microorganisms. The development of synthetic methods has helped in synthesizing some of the compounds that were originally discovered as products of microorganisms. Also, some purely synthetic compounds have antibacterial activity. Therefore, definition of antibiotic has been modified. An antibiotic now refers to a substance produced wholly or partially by chemical synthesis, which in low concentration inhibit the growth or destroy microorganisms by intervening in their metabolic processes. The search for chemicals that would adversely affect the invading bacteria but not the host began in 19th century. Paul Ehrlich, a German bacteriologist, conceived this idea. He investigated arsenic based structures in order to produce less toxic substances for the treatment of syphilis. He developed medicines arsenophenamine known as salvarsan. Paul Ehrlich got Nobel Prize for Medicine in 1908 for his discovery. It was his first effective treatment discovered for syphilis. Although salvarsan is toxic to human beings, its effect on bacteria, spirochete, which causes syphilis, is much greater than on human beings. At the same time, Ehrlich was working on azodize also. He noted that there is similarity in structures of salvarsan and azodize. As AS double bond AS linkage present in aspharamine resembles the N double bond N linkage present in azodize, in the sense that arsenic atom is present in place of nitrogen. He also noted tissues getting colored by dyes selectively. Therefore, Ehrlich began to search for the compound which resemble the structure of azodize and selectively bind the bacteria. In 1932, he succeeded in preparing first effective antibacterial agent, Prontocell, which resemble in structure to the compound salvarsan. Soon it was discovered that in the body Prontocell is converted to a compound called sulfanilamide, which is real active compound. Thus the sulfa drugs were discovered. A large range of sulfonamide analogues were synthesized. One of the most effective is sulfapyridine. Despite the success of sulfonamides, the real revolution in antibacterial therapy began with the discovery of Alexander Fleming in 1929. Therapy began with the discovery of Alexander Fleming. Antibacterial properties of a penicillin fungus, isolation and purification of active compound to accumulate sufficient material for clinical trials took 13 years. Antibiotics have either sedal, killing effect or a static inhibitory effect on microbes. Few example of the two type of antibiotics are bactericidal, means killing the bacteria, penicillin, aminoglycosides, ofloxacin. Bacteriostatic means inhibitory, erythromycin, tetracycline, chloramphenicol. coal. Here given in the side is H.W. Florey and Alexander Fleming shared the Nobel Prize for Medicine in 1945 for their independent contributions to the development of penicillin. Now given below is the range of bacteria or the given microorganisms that are affected by certain antibiotic is expressed as its spectrum of action. Antibiotics which kill or inhibit a wide range of gram-positive and gram-negative bacteria are said to be broad-spectrum antibiotics. Those effective mainly against gram-positive and gram-negative bacteria are narrow-spectrum antibiotics. If effective against a single organism or disease, they are referred to as limited-spectrum antibiotics. Penicillin G has a narrow spectrum. Ampicillin and amoxicillin are synthetic modification of penicillin. These have broad spectrum. It is absolutely essential to test the patients for sensitivity, allergy to penicillin before it is administered. In India, penicillin is manufactured at the Hindustan Antibiotics in Pimpri and in private sector industry. Chloramphenicol, isolated in 1947, is a broad-spectrum antibiotic, rapidly absorbed from gastrointestinal tract and hence can be given orally in case of typhoid, dysentery, acute fever, certain form of urinary infection, meningitis, pneumonia. Vancomycin and ofloxacin are the other important broad-spectrum antibiotics. Dicetirizine is supposed to be toxic towards certain strains of cancer cells. Antiseptics and disinfectants. Antiseptics and disinfectants are also the chemicals which either kill or prevent the growth of microorganisms. Antiseptics are applied to the living tissues such as wounds, cuts, ulcers, and diseased skin surfaces. Examples furacin, soframycin, etc. These are not ingested like antibiotics. Commonly used antiseptic Dettol is a mixture of chloroxylenol, terpenol, bithenol. The compound is also called bithionol. Is added to soaps to impart antiseptic properties. Iodine is a powerful antiseptic. Its 2-3% solution in alcohol water mixture is known as tincture of iodine. It is applied on the wounds. Iodoform is also used as an antiseptic for wounds. Boric acid is in dilute aqueous solution is weak antiseptic for eyes. Disinfectants are applied to inanimate objects such as floors, drainage system, instruments, etc. Same substances can act as antiseptic as well as disinfectant by varying the concentration. For example, 0.2% solution of phenol is an antiseptic while its 1% solution is disinfectant. Chlorine in the concentrations of 0.2 to 0.4 parts per million in aqueous solution and sulfur dioxide in very low concentration are disinfectants. Anti-fertility drugs. Antibiotic revolution has provided long and healthy life to people. The life expectancy has almost doubled. The increased population has caused many social problems in terms of food resources, environmental issues, and employment, etc. To control these problems, population is required to be controlled. This has led to concept of family planning. Anti-fertility drugs are of use in this direction. Birth control pills essentially contain a mixture of synthetic estrogen and progesterone derivatives. Both of these compounds are hormones. It is known that progesterone suppresses ovulation. Synthetic progesterone derivatives are more potent than progesterone. Northindrone is an example of synthetic progesterone derivatives most widely used as anti-fertility drug. The estrogen derivative which is used in combination with progesterone derivative is ethinyl estradiol, novestrol. Chemicals in food. Chemicals are added to food for their preservation, enhancing their appeal, adding nutritive value in them. Main categories of food additives as food colors, flavors and sweeteners, fat emulsifiers and stabilizing agents, flavor improvers, anti-stalling agents and bleaches, antioxidants, preservatives, nutritional supplements such as minerals, vitamins and amino acids. Except for chemicals, of category 7, that is nutritional supplements, none of the above additives have nutritional value. These are added either to increase the shelf life of stored food or for cosmetic purposes. In this section, we will discuss only sweeteners and food preservatives. Artificial sweetening agents. Natural sweeteners, that is sucrose, added to calorie intake, and therefore many people prefer to take artificial sweeteners. Orthosulfobenza- Orthosulfobenzamide, called saccharin, is the first popular artificial sweetening agent. It has been used as a sweetening agent ever since it was discovered in 1879. It is about 550 times as sweet as cane sugar. It is extracted from the body in urine unchanged. It appears to be entirely inert and harmless when taken. Its use is of great value to diabetic person and people who need to control intake of calories. Some other commonly marketed artificial sweeteners given in the table as aspartame, saccharin, sucralose, Elitame. Aspartame is the most successful and widely used artificial sweetener, roughly 100 times as sweet as cane sugar. It is methyl ester of dipeptide formed from aspartic acid and phenylalanine. Use of aspartame is limited to cold food and soft drink because it is unstable at cooking temperature. Elitame is high potency sweetener. Although it is more stable than aspartame, the control of sweetness for food is difficult while using it. Sucralose is dichloro derivatives of sucrose. Its appearance and taste are like sugar. It is stable at cooking temperature. It does not provide calories. Food preservatives. Food preservatives prevent spoilage of food due to microbial growth. The most commonly used preservatives include table salt, sugar, vegetable oil, sodium benzoate, phenoxide and sodium benzoate, and limited quantities and metabolized in the body. Sodium benzoate is used in limited quantities and is metabolized in the body. Salts of sorbic acid, propanoic acid are also used as preservatives. Antioxidants in food. These are important and necessary food additives. These help in food preservation by retarding the action of oxygen on food. These are more reactive towards oxygen than the food material which they are protecting. The two most familiar antioxidants are butylated hydroxytoluene and butylated hydroxyanisole. Addition of butylated hydroxyanisole to butter increases shelf life from months to year. Sometimes butylated hydroxy toluene and butylated hydroxyanisole along with citric acid are added to produce more effect. Sulfur dioxide and sulfite are useful antioxidants for wine and beer, sugar syrups and cut, peeled and dried fruits and vegetables. Next one is cleansing agents. Cleansing agents, we learn about detergents. Two type of detergents are used as cleansing agents. These are soaps and synthetic detergents. These remove these improve cleaning property of water. These help in removal of fats which bind other materials to the fabric or skin. Soaps Soaps are the detergents used since long. Soaps used for cleaning purposes are sodium or potassium salt of long chain fatty acids. Example stearic, oleic, and palmitic acids. Soap containing sodium salts are formed by heating fat, glycerol esters of fatty acid, with aqueous sodium hydroxide solution. This reaction is known as saponification. In this reaction, esters of fatty acid are hydrolyzed and the soap obtained remain in colloidal form. It is precipitated from the solution by adding sodium chloride. The solution left after removing the soap contains glycerol, which can be recovered by fractional distillation. Only sodium and potassium salt are soluble in water and are used for cleaning purposes. Generally, potassium soaps are so soft to skin than sodium soaps. These can be prepared by using potassium hydroxide solution in place of sodium hydroxide. Type of soap. Basically, all soap are made by boiling fat and oils with suitable soluble hydroxide. Variations are made by using different raw materials. Toilet soap are prepared by using better grade of fat and oil and care is taken to remove excess alkali. Color and perfume are added to make these more attractive. Soaps that float in water are made by beating tiny air bubbles before their hardening. Transparent soaps are made by dissolving soap in ethanol and then evaporating excess solvent. In medicated soaps, substances of medicinal values are added. In some soaps, deodorants are added. Shaving soap contains glycerol to prevent rapid drying. A gum called rosin is added while making them. It forms sodium rosynate which lathers well. Laundry soap contains fillers like sodium rosynate, sodium silicate, borax, and sodium carbonate. Soap chips are made by running a thin sheet of melted soap onto a cool cylinder and scraping off the soap in small broken pieces. Soap granules are dried miniature soap bubbles. Soap powders and scoring soap contain some soap, a scoring agent such as powdered pumice and finely divided sand and builders like sodium carbonate and trisodium phosphate. Builders make the soap act more rapidly. The cleaning agent been discussed in Unit 5. Why do soap not work in hard water? Hard water contain calcium and magnesium ions. These ions form insoluble calcium and magnesium soaps respectively when sodium or potassium salt are dissolved in water. These insoluble soaps separate as scum in water and are useless as cleaning agent. In fact, these are hindrance to good washing because the precipitate adheres onto the fiber of the cloth as gummy mass. Hair washed with hard water looks dull because of the sticky precipitate. Dyes does not absorb evenly on cloth washed with soap using hard water because of this gummy mass. Synthetic detergents. Synthetic detergents are cleaning agents which have all the property of soap but which actually do not contain any soap. These can be used both in soft and hard water as they give foam even in hard water. Some of the detergent give foam even in ice cold water. Synthetic detergents are mainly classified into three categories: anionic detergents, cationic detergents, non-ionic detergents. Anionic detergents are sodium salt of sulfonated long chain alcohol or hydrocarbon. Alkyl hydrogen sulfate formed by treating long chain alcohol with concentrated sulfuric acid are neutralized with alkali to form anionic detergents. Similarly, alkyl benzene sulfonates are obtained by neutralizing alkyl benzene sulfonic acid with alkali. In anionic detergents, the anionic part of molecule is involved in cleaning action. Sodium salt of alkyl benzene sulfonate are an important class of anionic detergents. They are mostly used for household work. Anionic detergents are also used in toothpaste. Cationic detergents. Cationic detergents are quaternary ammonium salt of amines with acetates, chlorides, or bromides as anions. Cationic part possess a long hydrocarbon chain and a positive charge on nitrogen atom. These are called cationic detergent. Cetylmethyl ammonium bromide is a popular cation detergent and is used in hair conditioner. Cationic detergents have germicidal properties and are expensive, therefore they are of limited use. Non-ionic detergents. Non-ionic detergents do not contain any ion in their constitution. One such detergent is formed when stearic acid reacts with polyethylene glycol. Liquid dishwashing detergents are non-ionic type. Mechanism of cleaning action of this type of detergent is same as that of soaps. They also remove grease and oil by micelle formation. Main problem that appears in the use of detergents is that if their hydrocarbon chain is highly branched, then bacteria cannot degrade this easily. Slow degradation of detergents lead their accumulation. Effluent containing such detergents reach the river, pond, etc. These persist in water even after sewage treatment and cause foaming in river, pond and streams, and their water get polluted. These days, the branching of hydrocarbon chain is controlled and kept to minimum. Unbranched chain can be biodegraded more easily and hence pollution is prevented. This is the summary of the chapter. Chemistry is essentially the study of materials and the development of new material for the betterment of humanity. A drug is a chemical agent which affect human metabolism and provide cure from ailment. If taken in doses higher than recommended, these may have poisonous effect. Use of chemical for therapeutic effect is called chemotherapy. Drugs usually interact with biological macromolecules such as carbohydrate, protein, lipids, and nucleic acid. These are called target molecule. Drugs are designed to interact with specific targets so that these have least chance of affecting other targets. This minimizes the side effect and localize the action of drug. Drug chemistry centers around arresting microbes and destroying microbes, preventing the body from various infectious diseases, releasing mental stress. These drugs like analgesic, antibiotic, antiseptic, disinfectants, antacids, tranquilizer are used for specific purposes. To check the population explosion, anti-fertility drugs have also become prominent in our life. Food additives such as preservative, sweetening agent, flavors, antioxidants, edible colors, and nutritional supplements are added to the food to make it attractive, palatable, and add nutritive value. Preservatives are added to food to prevent spoilage due to microbial growth. Artificial sweeteners are used by those who need to check the calorie intake or diabetic or want to avoid taking sucrose. These days, detergents are much in vogue and get preference over soaps because they work even in hard water. Synthetic detergents are classified into three main categories, namely anionic, cationic, and non-ionic, and each category has specific uses. Detergents with straight chain of hydrocarbon are preferred over branch chain as the latter are non-biodegradable and consequently cause environmental pollution. Thank you.